Hello there, all my dear students. We are passing through very strange and hard times, but please never lose hopes. During these times of self isolation, try to focus on pending assignments. Utilize your time very wisely. Especially, I would recommend to complete the assignments of biopharmaceutics and hospital pharmacy. Please stick to all the hygiene practices regarding coronavirus. Educate yourself, educate your community, and if people need any sort of guidance, please go ahead and let me know. I will appreciate your feedback, which you can give in the comments section. Lecture is about protein binding, so let's dive into that. So we discussed many times that there are all sorts of proteins in our body. These could be plasma proteins, these could be within the tissues like the receptors, or these could be macromolecules, maybe sometimes within the cells like DNA, melanin, and etc. So when the drug interacts with the receptor proteins, that leads to pharmacodynamics, as it could lead to possible pharmacological response, but it is not always the case. Sometimes it might lead to tissue damage or toxicity as well. So anyways, when a drug enters the body, it may interact with various sorts of proteins depending upon its nature and affinity, which is specific. Like all the weakly acidic drugs got affinity towards albumin proteins. If that gets saturated, then drug binds to lipoprotein. But the basic drugs bind preferably with alpha-1 acid glycoprotein. Here in the diagram you can see that as soon as the drug entered the blood circulation and as it comes into contact with the protein which it got the affinity towards as well, both will bind. So many drugs will interact with the plasma or tissue proteins or with other macromolecules such as melanin and DNA to form a drug macromolecule complex. Drug protein binding may be a reversible or an irreversible process. So why the drug protein bindings is sometimes irreversible? Because it involves the chemical activation of the drug. It might convert into a chemically reactive intermediate, and now that intermediate is ready to interact with proteins or macromolecules by chemical bonding. The bond will be a strong covalent bond. So here we can see that the necessary step involved is formation of reactive intermediate first, and then a complex is formed through a covalent bond. The irreversible drug protein binding might cause toxicity that could be long term, like the chemical carcinogenesis, or it could be short term. As you can see in this diagram, if you remember, we discussed about the paracetamol producing a reactive intermediate with the name of N-acetyl parabenzoquinone imine, which is the encircled one in the diagram. And it is formed as a result of phase 1 reaction where the paracetamol gets subjected to oxidative reaction. If, for example, it doesn't find the glutathione, then as shown by the red arrow, then definitely it makes a covalent bonds with the liver proteins that leads to necrosis, as shown on the right side of the diagram. I hope it is clear now that how this irreversible binding of reactive intermediate of paracetamol is reacting with liver proteins and causing hepatotoxicity. As far as the pharmacokinetics is concerned, the reversible protein binding is of high interest. It is called reversible because it doesn't involve the covalent bonding, but weaker chemical bonds. Hydrogen bonds or van der Waals forces are the best example for that. Actually, amino acid molecules that constitute the protein molecule have hydroxyl carboxyl sites available for such interactions. as you can easily see in this diagram. Once such a protein-bound drug complex is formed, it is such a large complex that it cannot easily transfer cell, any capillary membrane, and therefore it has a largely restricted distribution.